Put it in your pocket.
Yeah, perfect. Thank you. I'm so much excited down here.
Rikke's going to talk to us a bit about her research.
Correct?
Yes.
Thank you, and thank you very much for inviting me. I'm really excited that I get to share some of my research here with you today. I've chosen to, this is annoying, I've chosen to share some research with you that I did during my PhD because I think it's almost science fiction and I really like science fiction. Right now I'm a postdoc at the infectious disease department at Skyby Hospital. But what I'll be talking about today is something with humanized mice and HIV, and more specifically how we can understand HIV prevention and study it better with the help of this model. So first, what is HIV? It's pretty much in the name. It's human immunodeficiency virus. It's a virus. That's not a surprise to most of you, I hope. It can only infect humans, and I've chosen a nice gender-neutral female-male model here in this special day. The way a human can become infected is normally through body fluid, sexual transmission, or blood needles, drug addicts, or blood transfusions. The last thing is what it does. It causes immunodeficiency, and basically that's a defect immune system. If you have a defect immune system, you are going to have frequent and serious infections, often from pathogens that won't actually be a pathogen to normal people. And you will also get certain cancers that are caused by other viruses. And this whole thing here is actually called AIDS, Acquired Immunodeficiency Syndrome. And this is what it was named when it first was discovered in the 80s. Back then they didn't know it was a virus, so they just called it AIDS when they saw this combination of infectious and cancers. Then later they found out it was actually a virus and they could name it then. So why are we still interested in it? And it's not so much in the news anymore. But actually there's still there is about 37 million people that are currently infected with HIV. Out of these, only about 25 million people know they have HIV. And again, out of these, only about 20 million are getting the correct treatment. And out of these, only 60 million are getting fully correct treatment so that the virus is actually completely kept at bay in that person. And there's about 1 million deaths per year. More importantly, there's about almost 2 million new infections every single year. So this is still ongoing, and if we can't deal with it, then the circle is going to get bigger and bigger. So how can we actually prevent new infections? And we'll get back to that. But first I want to tell you a little bit about what does HIV actually does when it's inside the body. So there's two things you need to know. One, the left one here is HIV, the virus. And then we have a cell, it's an immune cell. It's called a helper CD4 T cell. And this is the cell that HIV infects. And it infects the cell, it replicates inside the cell using the cell's own system for creating new viruses. Then all of these viruses come out from the cell, and in the process the cell dies. And then we have more virus that is ready to infect new cells in the body. And then this cycle goes on and on. So why are these called helper cells? This is important for why HIV does what it does. So this is a very brief schematic overview of our immune system. We have different cells. It doesn't matter so much what they're called. The important thing is that this helper CD4 T cell has the role of coordinating and helping the function of all these other cells. So if the CD4 T cell, the helper cell, is not functioning properly or is not there, then everything kind of gets messed up. Another thing, something that's called killer, is something we're interested in when we have a disease, a cancer or an infection. So I just want to highlight this cell because I'll be talking a little bit about it later, because this is a cell that can actually kill HIV and HIV-infected cells. Good. So how does an HIV infection look like? So here we have the black line here. That's my pointer. No, mine. Well, the black line stands over at 0. That's when a person becomes infected. Usually they don't know it in the beginning. And then you can see the black line is the virus. So the virus grows in the body at an exponential rate. And already within a few weeks, you have very high levels of virus in the blood samples of this person. And then in the beginning, and the immune system can still kind of keep the situation in check. And actually the immune system is able to suppress the virus. As you can see, the black line goes down. And for a number of years, everything is kind of okay. Even if you don't know you have it, the virus is kept down and the immune system is still kind of functioning. Then later, it has become too much. And at that point, the virus is going to start replicating more. This is when those helper CD4 T cells, this is the blue line, you can see it goes slowly down as the years go on. And once you've lost enough of your helper CD4 T cells, then the immune system can no longer function properly and keep the virus in check. Then it grows uncontrolled, and then this is when you will start to get these other infections and the cancers and eventually die from these other diseases. But luckily, there's been a lot of research done in this area, and we have very, very good treatment today. So if a person is diagnosed with HIV, they will usually, if they live in Europe at least, get the proper treatment. And this treatment can actually suppress the virus. So you see the black line is going down, and it's so efficient that if you take the blood sample from the person, you cannot even measure the virus in the blood, except if you have very special ultra-sensitive assays. So when you can suppress the virus like this, then you will not lose your helper CD4 T cells. And that basically means that you can live a normal life. And actually, we do say today that HIV that's well treated, it's more of a chronic disease than it is a deadly disease when you get the treatment. Of course, it's expensive, there are some side effects, and it would be better if we could just stop new infections from happening. So, what are the ways that we could possibly do this? Like other diseases, if we could get a vaccine, that will be completely optimal. Unfortunately, that is proving quite challenging because HIV is a very diverse virus. It changes fast. So there have not been any real good vaccine test results. There's also something called a micropicide, which is basically, it's a gel with the HIV medication inside. And then you put it in the vagina or the rectum so that when you have sex, if your partner is infected, then that drug being present there would hopefully protect you from becoming infected. This has also had very variable results. Some of them have shown not to work at all, some have worked a little bit. One thing that's actually very interesting, it has been shown that one of the drugs is metabolized by the different bacteria in the vagina, and differently metabolized by different bacteria. So the woman, if a woman had, for example, bacterial vaginosis, which is an overgrowth of the not so great bacteria, then the drug will get metabolized much faster, and it actually won't have the preferred effect. So, and this is something they found out after doing massive, huge, expensive studies testing these micropicide, and they didn't understand why the studies had such different results. And then they discover afterwards, oh, but if the vagina flora is not what it should be, then the results are completely different. So we learn things all the time, but it just shows you need to know the environment you're putting something into if you must know the full effect. Then there's pre-exposure prophylaxis, and this is where you treat an uninfected person if they're going to have a risky behavior. So that can be a couple, for example, one is infected, one is not, and one is not on treatment yet, something, or you're going to travel to Thailand and be a little wild. Then it's actually, I'm not kidding, it's actually been approved that if you go to your doctor and say, I'm going to have such and such behavior, can you please prescribe me some pre-exposure prophylaxis, that is allowed. I wouldn't recommend it. So all of the first three versions here were about treating people that are not infected in order for them not to become infected. So there is another option, which is treating the people that are infected in order to protect the people that are not infected. And we call it HIV treatment as prevention. It may not seem surprising, it's pretty logical. If you treat a disease, then you can't pass it on as easily, but we like to prove things in science. And in 2011, there was a big trial where they had like 1,700 couples where one was infected and one was not. And they proved that if you chose to treat the infected person earlier than you normally would, then actually the partner was 96% protected. So this is a very efficient way of protecting the uninfected partner if the other one is infected. And this was actually the first time it was proven that you can treat someone and then protect the other one. You can't, however, really go around treating everyone in the world for them not to become infected. That would be really expensive and unpractical, probably also a little unethical. But it did now, it worked, and the hypothesis is, of course, that when you give HIV treatment, then the levels of HIV in your genitals, so in the vagina and in the semen, are actually too low that it can transmit to another person. So in the clinical study, they didn't look at this, they just did blood samples because it's a massive study. And here's how it would look like if you drew it. So here's again my no gender man. And this is a person that's infected but not on treatment, and there's a lot of virus all over the body here. And of course, it's in the blood, it's in the entire body, and then it's also in the genitals, and you have a high risk of infecting your partner. So here's a person on treatment, and then you have low levels of HIV in the blood, also in your genital secretions, and then the idea is you have low risk of infecting the partner. But like I just mentioned before, in these big clinical studies, it's really hard to get genital secretion samples. It takes a special dedication from the patients to deliver these samples. Let's just say that. So we would like to know more about how it actually works. So the project that I had, among other things, for my PhD was to understand how can this HIV treatment prevent HIV transmission, especially in the context of the level of HIV and also the immune cells in the vagina, because the immune cells are what becomes infected by HIV. So that was my project there. And how are we going to go about doing that? So mice. Mice is a great model to investigate many different diseases. But like I showed you in the beginning, HIV can only infect human cells. So we cannot really just put HIV into a normal mouse and then nothing is going to happen. It won't replicate in there. Humans, yes, they can become infected. There is an ethical issue giving people HIV. It's also not legal. You could, of course, then take some patients that are already infected. The real challenge is that most people don't know they're infected for at least a couple of months, usually a couple of years. So how are you going to get the samples from their vagina right after they had sex and became infected? Tricky study, very tricky. And then we have all of these monkey models, which some of you probably have heard about. And they are great models for many things, but the reality is you cannot infect them with HIV. There is a different virus that's called SIV, simian immunodeficiency virus. And that one you can use in these monkeys, but it's not completely similar to HIV. So if you really want to study HIV, the human version, then you have to use human cells. So no mouse, no monkeys, no human. What do we do? Someone thought of this and said, let's make a humanized mouse. A humanized mouse can be many things. It's basically a mouse that has some kind of human component in it. In our research specifically, it is a mouse. It is a mouse that actually has a human immune system. Why is that great? That is, of course, great because then we have the helper CD4 cells there, and that means we can give it HIV. So now we have a small animal model that we can infect with the HIV. So how do we make these mice? I'm going to give you the basics. So there are quite a few different ways to make these mice. What's common for all of them is that you need a mouse that does not have its own immune system. This is because if you think about, if you need a kidney transplant or a blood transfusion or something, then you need your tissue match. You need a donor that has the same tissue type as you do, because if you don't get that, then your immune system is going to attack whatever you put into the person's body and reject it. The same thing would happen if you put something from one species into another. So we cannot just put something human into a normal mouse with a functioning immune system. And then we need some human immune stem cells. So we get this mouse here. Luckily, someone breeds different kinds of these mice with no immune system. And then we get some human stem cells. We inject them into the mice, just a tail vein injection. We wait about 8 to 12 weeks, take good care of them. We take frequent blood samples from these mice to measure then how is it going with these human cells. And then usually about those 8 to 12 weeks, we have a mouse, we take a blood sample and we can measure these different kinds of cells in there, including the human CD4 helper cells. And that means the mouse is now ready to have an experiment performed with it. So what happens when we give HIV to the mice? This is the same graph I showed you before, so that's from humans, where you have the virus is the black line and the helper cells is the blue line. So this is from a mouse experiment, and again, the virus is the black line, and you can see the virus grows very nicely in these mice. This is over 6 weeks here. And also, like in the humans, the blue line, you can see you get less and less helper CD4 cells in the mice. So in this way, the course of the infection is somewhat similar to what you see in humans. We can't really keep the mice alive for years and years, so the time span is a little bit shorter. What I just said. So, going back to what we really wanted to investigate was what happens in the vagina. So next we want to look at, is HIV actually in the vagina of these mice? And yes, I washed a lot of mouse vaginas during my PhD. It's not very exotic or thrilling, but it gives nice data. So the black line here, it's again, that is the virus in the blood for comparison. And the dashed line with the white dots, that's actually the level of virus in the vagina. And you can see already very shortly after we expose the mice to HIV, we have a lot of HIV in that vagina. And even before it's even detectable in the blood. So it indicates that a lot of replication is going on locally there in the beginning already. So that's good. What happens to these immune cells that we talked about? So this is immune cells that's from the washing of the vaginas. And now I showed you before that actually you lose your CD4 helper cells when you become infected. But what you see here is very interesting. The blue line is again the helper cells, and it increases. That's not very good for us, because that means HIV has lots of targets to replicate in. That's why it can grow exponentially and very fast in the beginning. It's also showing the red line. The red line are those killer cells that could actually kill infected cells. And the red line also decreases, but it increases later than the helper cells. So the virus has a good head start with lots of target cells before the killer cells come in and has a chance to control the infection. This is also some hypothesis that's been shown in monkey studies. But it really gives us an understanding of how quick we need to be if we need to prevent the virus from establishing itself in the body. So this is all what happens without the treatment, and we wanted to see what happens with the treatment. And now again, this is the virus in the blood and in the vagina, and the gray area is when we actually gave the mice HIV treatment. And you can see it looks very nice. There's a lot of virus before we give the treatment, and then already 2 weeks after we start the treatment, the virus is almost undetectable in both the blood and the vagina. And again, when we look at the immune cells, you can also see here you have CD8 cells coming in, you have the helper cells coming in. And as soon as you start the treatment, then it goes down and the levels become somewhat normal. So you kind of normalize the situation by giving this treatment here. And if we go back now, this was the hypothesis created from the clinical study. It was that HIV reduces genital HIV levels. Well, yes it does. In the human mice at least, so probably also in humans. And this is what we saw. HIV treatment, it suppresses the HIV in the vagina and in the blood. We already knew that. And it also restores the immune cell numbers to normal. So for once in science, which is not the norm, we have a conclusion that supports the hypothesis that we started out with. So that's a nice feeling, if you haven't tried that yet. Yeah, so this is the main conclusion from the study that we did. And going back to the real life, not with mice, if we look at this circle again now, here, this top part here, if you deduct the numbers from each other, you basically have 17 million people that are not on treatment. These people are infecting other people. So what's really, really important if we want to prevent new infections is how do we find these people? And there are a lot of big studies and effort in what we call seek and treat. So how do we find people at risk fast and get them on treatment so that we can somehow break this infection circle here? Yeah. And this is just briefly the people that helped with the study, my colleagues. Don't remember if I mentioned, but I did this work in my PhD. It was in North Carolina at University of North Carolina. And so this is all my colleagues over there. And lastly, the last year and a half or so, we have actually been working to set this model up here in Denmark, because you can do some pretty cool stuff with it. And it is working now, and we are getting ready to perform our first HIV experiments. So yay! And that's that.
All right, thank you, Rikke. Do we have any questions? I can take one. Yeah? Yes.
So this might be a bit out of scope, but is there any plans for doing correlation between the genetics of the stem cells used and the susceptibility to HIV?
Well, let me answer it in a different way. Not correlation. You have to know specifically what you're looking for. And there have been done experiments. So there's one specific gene that does so that the helper cell, the receptor that the virus uses to get into the cell, we know that people that have a specific mutation in this receptor, if they cannot become infected with HIV. And if we get stem cells from these people and put it into the mice, then the mice also do not become infected with HIV. So whatever genetics it's in the stem cell, it's gonna be in the mouse immune system. That answers.
Okay, thank you.
Thank you.